Okay, so this little lecture is to introduce you to some of the techniques used in the Staphylococcus epidermidis paper that describes a anti-cancer compound produced by this specific strain of Staph epi. So this is just a picture. Remember, Staphylococcus means circular bacteria that grows in clusters. Epidermidis tells you that it grows, is found on the skin. So it is right here. It's an aerobic bacteria. They talk in the paper about it being coagulase negative. So they're just explaining a little bit of the characteristics to you. It is part of our normal flora, so it's really easy to isolate. When I used to teach a micro lab that didn't have very many specimens, I would just take a Q-tip, rub it behind my ears, rub it on agar, and get these lovely Staph epi colonies. So it's all around you. It helps protect your skin from other pathogens. It has some communication with the immune system, and like I said, they're finding that some strains of it produce a molecule called 6-HAP, which has anti-cancer properties. And so that's what they're going to talk about, and that's what they're going to characterize in this paper. So just to remind you about cancer, cancer is basically uncontrolled cell division. And so I put an image of the cell cycle to remind you that as cells divide by mitosis, they really go through this whole cell cycle of growth, DNA replication, more growth, and then the fun mitosis where you see the separation of the chromosomes. In cancer, in cell biology, in general biology, we talk about checkpoints at different phases of the cell cycle that slows the cell down, goes through, checks, makes sure the DNA is replicated correctly, make sure the spindle fibers are set up, make sure there's enough material to go and divide. And what happens in cancer is that you lose some of these regulatory mechanisms, and the cells will just continue to grow and divide and grow and divide. And what a lot of times happens is because the loss of the checkpoints, DNA replication becomes full of errors, right? And errors in the DNA sequence can lead to mutations in genes. So this is at a molecular level. We are going to talk a lot about DNA replication, so that's during S phase of the cell cycle. At the cellular level, you start out with a normal cell that gains a mutation, and somehow this mutation allows it to continue to go through the cell cycle without stopping at all the checkpoints. And one of the hallmarks of cancer is that the cells don't have what we call contact-dependent inhibition, where normally cells make these nice layers, and when they bump into their neighbors, they know there's no more room, so they stop dividing. But cancer cells will just start piling up on each other. They can grow in different types of tumors, so we're going to see some that are these big tumors and some called papillomas, which are kind of more tight, like a wart tumor. And then, unfortunately, especially with melanoma, one of the reasons it's such a deadly type of skin cancer is that this cancer can spread to other parts of the body, and that's called metastasis. And though we don't really go into that in this paper, I just want you to understand that that's why skin cancer can be so deadly, is not that it's just affecting your skin, but pieces of it can break off, move through the blood vessels, blood system, and start making cancer in other organs of the body. So what is 6-HAP? It's this molecule shown right here. Looks like the base adenine, right? So remember DNA, you have A, G, C, T as your bases, or the things that make your nucleotides. So it has this purine ring structure, lots of nitrogens there, and so that it looks like adenine. Okay. The problem is, and it's, sorry, I want to make the word, you'll see this word, it's an analog because it has structural similarity, but it doesn't necessarily act the same as adenine. So remember in DNA, A's and T's base pair, and the way they form stable base pairs is by two sets of hydrogen bonds, which are shown in the red dotted lines there. Okay, so that's important. So when you're replicating your DNA and making a new strand, right, you're going to copy, you're going to put a G in here, and then you're going to copy, and you're going to put a C in here, and you hook them together. And then you're going to copy and put, oops, an A, hook it together, copy, put a T, hook it together, right? So you're synthesizing 5' to 3'. Okay, so if 6-HAP gets incorporated into a nucleotide, when the DNA polymerase is trying to match it, like it would be right here, to adenine, so this is the DNA, this is the new strand, I should write parent, it can't make the proper hydrogen bonds. So if it can't make the hydrogen bonds, the DNA polymerase just hangs out there and is like, I don't know what to do. And so that's how 6-HAP can inhibit, DNA synthesis. Okay, so because it doesn't make the proper base pairs, the DNA polymerase gets stuck, and the DNA polymerase that I'll talk about in a minute that they use in this paper to test it doesn't have proofreading, so it really just gets stuck and just sits there and stops DNA synthesis. Why is this important for cancer? Well, if cancer is uncontrolled cell cycle and there's lots of DNA replication, if you block DNA replication by adding this molecule 6-HAP, then you can't continue cell cycle. So the tumor can't grow. Right? So this is where 6-HAP is working, right there at this step of synthesis in cell cycle and blocking DNA replication. All right, so how do they characterize 6-HAP? So they do a lot of chemistry to identify, and purify, purify the 6-HAP molecule. So they do things like high performance liquid chromatography shown here. They do mass spec. They do proton nuclear magnetic resonance. So we're not going to go through these specific chemistry procedures. The whole point is so that they can directly identify the atoms involved in making 6-HAP, how it's arranged, and that they can purify it. Remember that eventually this is synthesized in the lab. which means it's artificial. So first they do some purification from the bacteria, but I'm trying to remember where I wrote this. They only get like 7 milligrams. Where did I write it down? I never can find anything when I want to. Something like 7 milligrams per 6.4 liters. So that's a lot of bacteria you have to grow up for a little bit of this compound. So it's great that it can be synthetically made in the lab, because if we want to use this for a drug, we have an easy way to mass produce and produce it very purely. So you don't have to get rid of other contaminants from the bacterial cells. Another way they characterized 6-HAP is looking at zones of inhibition. So if you remember this from your microbiology, this clearing equals no growth, no visible growth of the bacteria. So remember, to do this, you take an agar plate and you spread bacteria all across it to get a lawn. And then on these little punches, you add your antimicrobial agent, which in this case would be 6-HAP. And this antimicrobial agent diffuses from the little hole punch out into the media, and as this lawn of bacteria is trying to grow, depending on the strength of the compound, it will inhibit growth. So you're looking for clearing, meaning you're inhibiting bacterial growth. They also in the paper talk about colony forming units, and this is a way to measure what I'm writing is alive bacteria. So they also look not just on a plate, but they actually mix the 6-HAP with some group A strep. That's gas, just another type of bacteria, and they mix them together and then they plate, and you see how many colonies are formed. Remember, 1 colony equals 1 bacterial cell. So you can calculate how effective a compound is at killing or blocking growth of bacteria. So we talked about that 6-HAP works. The reason it blocks the growth of group A strep is that it's inhibiting DNA synthesis. So the way they show this is a DNA replication assay, and they use Klenow, and they write EXO minus. So Klenow is a DNA polymerase from bacteria. Okay, so DNA polymerases are involved in DNA replication, right? Exonuclease minus means there's no proofreading. So this is actually a mutant form of a DNA polymerase. So this polymerase, as it's copying, it can't go back, backtrack, remove a mismatched base, and continue on. So Klenow can only go and synthesize 5' to 3'. And so what they use is they use what we call a primer, and it's labeled, right? It's got a dye to it so we can visualize it. So you have a template strand, and then Klenow, the DNA polymerase, comes here and starts to base pair and make a complementary sequence. So the primer by itself, when you run it on a gel, has a band of a certain size, and the primer plus DNA replication, right, if it could continue, has a larger size. So you'll see some bands on figure 1F And so they're just looking at, if you add 6 HAP, does it interfere with DNA replication? Another type of DNA replication assay is using BrdU. BrdU is shown here is an analog of thymidine, so of your T nucleotide that goes into DNA. This does not interfere with DNA replication, but what it's trying to show you is as the DNA is replicated, you can get incorporation of this new nucleotide. And then what you do is you denature it, and there are antibodies that will recognize BrdU, bind to it. Excuse me. I'm having the worst allergies. Bind to it, and then you see a signal, so you can visualize. And so this is called an incorporation assay. So they're measuring how much BrdU is in new DNA, which really means how much new DNA is being made. Right, so it's just a way to say, yes, these cells are actually going through cell cycle and dividing and doing DNA replication. And as you see a decrease in BrdU incorporation, you know that that means less and less DNA is being synthesized. The gene expression assays and the siRNAs. Gene expression, I am guessing that they're looking at mRNA levels. So probably doing qRT-PCR, though I could not find it anywhere in the paper that says it. This part that says relative to GAPDH. GAPDH is called a housekeeping gene. Oh my gosh, my cat is outside my door howling. Hold on. Oh, shit. Come on. Sorry about that. You probably couldn't hear it, but it was distracting me. So GAPDH is a housekeeping gene, which means it's expressed at constant levels in cells. So it's just a control, and it's a way to keep things even. Okay? So you want all samples to be compared at the same amount. So I want to know if there's an increase in gene expression of a gene. I want to know it's not just because I accidentally added more sample to that experiment, but it's actually because it's higher, it's expressed more highly than in a different cell type. And so we normalize, that's the word I was looking for. We normalize the gene expression levels to the housekeeping gene, and then we look at the specific gene we're interested in and can decide if there's an increase or decrease in the level of expression. siRNAs, your short interfering RNAs. We kind of talked a little bit about these when we read the paper with microRNAs. An siRNA, in this case, is a synthetic sequence, so they make it in the lab, that's complementary, that's just a pain. Synthetic sequence complementary to, I know my pen's not going to work, to your target mRNA. oops. So if I want to knock down the mitochondrial ARC2 gene, then I make a siRNA that's complementary to it. That small RNA associates with this RISC complex, and it cleaves, chops up the messenger RNA. So you get a decrease in expression. So the siRNAs are very specific to the different genes that you want to knock down. And we call it a knockdown because it's not 100%. Once you've made messenger RNA, there's always a chance that it can get translated before the RISC complex targets it. But what you can see here, maybe, is that siRNAs to ARC1 decrease ARC1 and not ARC2, and siRNAs to ARC2 decrease ARC2 and not mARC1. Right, apparently my slides want to go on. There's also a couple mouse models involved. Isogenic melanoma tumors. Melanoma is the skin cancer. And what they do is they insert cancer cells right under the skin, right under the epidermis. Okay, so they're taking cells from another place. Okay, these aren't the mouse cells. These are actually cancer cells that they grow in tissue culture. They inject it under the skin. Whoa, I don't know what that was. And then they let it grow and it will form tumors. And so this is one of the figures from your book showing this lump. So this is a big tumor that's grown from these cells that they've implanted under the skin of these mice. The second type of cancer model they use is called the two-stage UV carcinogenesis model. So they use these little hairless mice. Okay, so you don't have fur, hair interfering with the ultraviolet treatment, and they first rub something called DMBA on the skin, and this is a carcinogen, which means it causes mutations during DNA replication, and DNA mutations can lead to cancer. So one, they give them this chemical. Two, they UV irradiate, so you know that if you're out in the sun getting hit by the UV rays from the sun, you have a chance of the ultraviolet light mutating your DNA, causing thymidine dimers, and that can also lead to cancer. So now they're inducing cancer on this little hairless mouse, and the types of cancer they're seeing here are papillomas, which are like warts. Okay, so it looks different than this big blob tumor. There are these distinct tumors, and one of the nice things about this model is that you can count them, right, because they make these distinct papilloma tumors. So papilloma just describes, looks like a raised bump, looks like a wart. Okay, I think my last figure for you is there is a heat map. They're screening to see from different people, different subjects, who has staph epi strains that express 6 HAP because not all strains of Staphylococcus epidermidis express that, and so they're looking at different body parts versus the different subjects. And the heat map means the more yellow, the greater the abundance of Staph epi strains that produce this, and blue is a much lower abundance, right? So if you had to, right, you could go to someone's palm or maybe their heel and have a pretty good chance of getting one of these strains. Okay? These people with all blue, or mostly all blue, mean they don't have strains of Staph epi that produce the 6 HAP. So as you work for the next 2 weeks, this is your last paper. As a group, you're going to go through each figure. You want to make sure you understand the experiments, how they were done, why they were done, talk about the results, which is the data, talk about the conclusions, which is the big picture. What does this mean? Why is this significant? And go ahead and discuss next steps that you would like to see from this paper. All right, that's all I have for you. I hope you enjoy reading this paper.